And also, once we identify the most relevant cells and a part of the tissue for human disease in humans, and I hope this can help us for better or precision disease treatment in the future. Yeah, so we often just go out to look at people who are affected and unaffected. So we have two populations, one population of affected people and another population of unaffected. Then the question is, how can we know what are the factors that drive the difference, right? So we have differences in lifestyles, but most importantly, we have differences in our genome, the DNA difference, right? So say we have seen a DNA variant in the population, and then we ask, what is the frequency of the DNA variant in the affected population, and what is the frequency in the unaffected? And then we compare the difference by a statistical test. If we see a real difference, then we claim, hey, this variant is associated with the disease. Then we do that repeatedly for many different variants across the whole genome. This process is called a gene mapping or map or genetic association study. So we are a team interested in developing methods to help people to do this kind of gene mapping more efficiently or more in a more powerful way. I just mentioned this genetic association study, right? Or genome-wide association study, or we call it GWAS. So GWAS can help us to identify the genetic loci or the genes that are relevant to a disease. But we don't know which part of the body where these genes exert their effects, or what are the cells, like precisely what are the cells? What are the cells that are relevant to a disease? Where are they distributed in our body, right? So this is a kind of challenge, and I felt really excited about this question. So I asked one of my students, can we really think about a model to do that? So initially, it was very difficult because there's no such kind of data allow us to kind of measure cellular level gene activity, right? Until this technology called spatial transcriptomics. So this is the kind of technology you can make a slice of a sample, typically an animal model. Once you make a slice, you can essentially look at not only the expression, sort of gene expression activity for any particular cell, you also have information about the cell's location, right? Because you keep the original location when you do the slice. So once you have this kind of data, so essentially this is the data where you have cells, gene activities. On the other hand, you have GWAS data, you have disease and a gene, right? Disease and gene link. And in this case, you have cell and a gene link. So we then came up with an idea to say, well, can we link these two kind of data sets by the genes? We have gene activity, cell distribution. You have disease and a gene. You can then link cell to the disease by the genes. So once you have that, we were able to identify what are the cells on this spatial transcriptomic data that are related to a disease. So there are two important applications. The first is to understand disease biology, right? So we, when we study depression, schizophrenia, this kind of psychiatric disorders, we often use animal models because we can't study human directly, right? So with animal model, we need to think about what are the most relevant cells we need to study in these models. So once we have a better, just to remind you that, so the method is very kind of general and flexible. It can map human disease genetic data or genome-wide association data for human disease to a spatial transcriptomic data for an animal. So it allows us to map human disease to animal cells. So once we do that, we can better understand what are the cells, what are the sort of tissue, specific region of the tissue that is good or bad for understanding disease biology in a mouse model or in any animal model. So this is the first application. So the second application, which I found really exciting about, is you can really use this kind of technology to help us for sort of precision medicine or precision treatment. So just to give you one example. So we use a monkey spatial transcriptomic data. That monkey is called macaque. So we have not a whole brain, just a cortex. So because the brain is too big for this technology to measure. And then you just have one single area, it's called cortex, but we have the whole cortex data. So when we map the human psychiatric disorder GWAS data to the macaque cortex spatial transcriptomic data, if we find a region which is very kind of relevant to depression, and we look at then further the region, why this region is showed most relevance to depression. We look at the expression level of the cells in that region or the gene expression activity. What are the genes that are most active in that region? We found that these genes, interestingly, have strong enrichment in existing human psychiatric disorder drug-targeted genes. So you have a gene highly active in that region that we identified in macaque cortex area. On the other hand, you have a lot of human psychiatric disorder drugs, right? And these drugs should have gene target. They target some genes. We show a strong overlap of these two set of genes. Amazing, right? Human psychiatric drug targets, and this is a macaque, a region in a monkey brain, and show some gene activity. So that kind of indicate or really encourage us to look further. Really, there's a possibility to have precision medicine, kind of precisely treat some specific cells for better treatment.